Assalamu alaikum. Welcome back for our next lecture. Today we shall discuss the concept of sequence similarity and identity, which is used in bioinformatics for understanding protein sequences of different sources. For DNA sequence, there is no difference between the terms of sequence similarity or identity. So therefore, when you are studying DNA sequences, you can use either sequence similarity or identity. But when we are studying protein sequences, there is a clear difference because sequence similarity is based on physiochemical characteristics of the amino acids that are being aligned. On the other hand, in DNA, in protein sequence identity, there is no concept of physiochemical properties. So when we are aligning two or more sequences of proteins, sequence similarity refers to the percentage of aligned residues that have similar physiochemical characteristics, which means you will calculate the total number of amino acids which are aligned together, which are aligned in the same column in sequence alignment, and they have similar physiochemical properties. Sequence identity is kind of, how to say, simple as compared to sequence similarity. For sequence identity, you will have to count only those amino acids which are identical. Or in other words, when same amino acid is present in multiple sequences that are aligned, it refers to as sequence identity. So let's define it. Sequence identity refers to the percentage of matches of the same amino acid residue between two aligned sequences. So let's discuss it with the help of an example. Here is a diagram representing sequence alignment of three sequences. We can see there are some gaps, there are some mismatches, and then we can see there are some sequences which are highlighted with different colors. So these different colors, they represent different groups of amino acids having similar physiochemical properties. For example, the gray color represents those amino acids which have aliphatic side chains in their structures. On the other hand, this yellow color represents those amino acids which have sulfur containing uh, functional groups. Similarly, there are some basic amino acids, acidic, and then there are amino acids which have different other physiochemical properties. Here we see an amino acid which is named glutamic acid. It is present in all the three sequences. So we can say it is similar. We can also say it is identical. So it is the same amino acid. But if we look here, there are two amino acids. One is glutamic acid and the other one is aspartic acid. So this column, the amino acids present in this column are not identical, but they are similar because these two amino acids, they are acidic in nature. So we can see similarly here, there are glutamic acid and aspartic acid. They are aligned in three different sequences, and we can say these are similar sequences. So I hope you have understood the concept of sequence similarity and identity. So here, for example, here it is an identical sequence, and these are the similar sequences. Now the question is it possible to calculate a total amount of sequence similarity or identity? Yes, we have several formulas that can be used to calculate the total amount of sequence similarity or sequence identity. In coming slides, we shall discuss two formulas. For example, this is a sequence similarity calculation formula. Here, S is the percentage sequence similarity. LS is the number of aligned residues with similar characteristics. And LA and LB are the total lengths of each individual sequences. For identity, uh, This formula is used where Li is the number of aligned identical residues. So these formulas are generally used for larger sequences. So sometimes we have very small sequences. So for smaller sequences, we have another formula, which is obviously relatively simpler. For example, here percentage similarity is equal to the formula where LA is the length of the shorter of the two sequences. So these two sequences, they have different lengths, and for identity Li over La. So where Li is the number of aligned residues with identical residues, and La obviously is the length of the shorter of the two sequences. So this is all for the concept of sequence similarity and identity. I hope you have understood the difference of these two terms, and it will be helpful for you in coming days. Thank you for your time and for your consultation.